welcome everybody to the launch of the new Midlands Innovation Immunology Network and our COVID-19 Research Symposium. We've got a really interesting afternoon of talks for you with a great mix of both national speakers and speakers from within the Midlands and attendees from all over the country. I thought I'd start by giving you some history as to how we got to today's event. As some of you may know, I'm chair of the Midlands Innovation Flow Cytometry Network. And whilst looking at the rebrand of the Midlands Innovation website, I noticed a section on Midlands Innovation Health, but immunology was not represented. So at the start of February last year, February 2020, I contacted Ellen McGee, who supports my role as chair of the Midlands Innovation Flow Cytometry Network, to discuss potentially starting a new immunology network. A couple of weeks later, Alex Archibald, who is supporting today's event, and she supports Midlands Innovation Health, then contacted me, and just before COVID-19 hit the UK, we met in person to talk about my thoughts. And Alex has shared that journey from that meeting back in February last year through to today and this launch event. So we set up this network with the ultimate aim to bring together immunology colleagues from across the entire Midlands region, complementing initiatives such as the British Society for Immunology, to facilitate discussions related to grant funding, and to be a hub to share technical expertise in the immunology arena. Our network is made up of senior colleagues at each institution within the 8 Midlands Innovation Universities to ensure we meet our aims and ambitions. This afternoon, we've got 4 sessions looking at different components of the immune response to SARS-CoV-2. One looking at antibody measurement, one examining cell-mediated responses to infection, one exploring the development of vaccines, and finally, one looking at the systemic immune responses to SARS-CoV-2. Each of our presenters has just 30 minutes to talk and then 2 minutes to answer 1 or 2 questions. So please, attendees, please enter your questions in the Q&A box within Zoom. We'll have a 20-minute convenience break after the first 2 sessions. And then after the last 2 sessions, we've organised breakout rooms. At this point, you'll actually need to change and go to the breakout room that you've registered for. And details on how to do this were emailed to you prior to the event. In these breakout rooms, you can go and join in scientific discussions on one of the 4 topics we will have covered this afternoon. These will hopefully give you the opportunity to ask further questions of our speakers who have kindly agreed to stay for these discussions. And because there is so much high quality research taking place across the Midlands, we're already planning a sister event to take place in about 2 months' time to give other colleagues the opportunity to present their research. I know we also have some key national speakers who'd like to present at the next event, so please watch this space. There will be a sister event coming in a couple of months' time. So finally, it's a big thank you to our 5 sponsors for today. So Beckman Coulter, Biorad, Fluidigm, Luminex and Miltony Biotech have kindly sponsored the event. So thank you for being so supportive during such challenging times. We're going to use this sponsorship money to build collaboration within this new Midlands Innovation Immunology Network. So thank you for sponsors, and I encourage our attendees to get in touch with these sponsors and explore how they can support your research. Also, a great big thank you goes to Alex Archibald, who's been an incredible support during the organisation of this meeting. And without Alex, this whole meeting would not be possible. So thank you very much, Alex, for all of your support. So this is a fairly brief introduction. I'm going to hand straight over now to Professor Mark Drayson from the University of Birmingham, who's going to chair the first session on antibody-mediated immune responses. So please enjoy the afternoon. Thank you.
Well, hello and welcome to this first session. It's about antibody measurement, and we have 3 great speakers. I guess back in March and a bit before, when we were all thinking, what are we going to do about SARS-CoV-2 as immunologists, and of course measure antibody, but antibody against which protein? And inevitably we wanted to measure it against most proteins, but of course the spike protein was by far the most important. And we had spike from different places in the world, but we went outside a region to Max Crispin to get our spike because it was properly made and glycosylated and all the rest of it. And Max will be telling us about this as soon as I shut up. Alex Richter, my colleague in the office next door, started designing various ELISAs and other platforms for measuring antibodies against a variety of proteins, and gradually found the right one, but at the same time collecting large numbers of samples, because to put antibody testing into clinical practice, you need a lot of samples from different patients who've had severe disease or hardly any disease, etc. And then our final speaker is Alexander Tarr from Nottingham, who's going to be telling us about differing patterns of antibody responses in severe SARS-CoV-2 infections. So without any ado, can I pass over please to Max in Southampton.
So thank you very much for that kind introduction. I'm just about to share my screen, and hopefully that will all be visible to everybody. There you go. So it's a great honour to be honorary Midlands for the day, but it's not just today. We've been working incredibly closely with Alex, who's here in the next talk, and colleagues across Birmingham on sort of setting up the antibody assays. I mean, I should be immediately clear that my role has really been on the antigen side. So I'm focusing here on the development of the sort of native mimetics. And you can see in the title of the talk, the glycobiology of SARS-CoV-2 viral spike mimetics. You might be asking, what has glycobiology got to do with this? Which would be a fair question. But you'll see from this image that actually a lot of these envelope viruses contain the viral spikes that are really significantly dominated by carbohydrates. So if you're interested in having native mimics, the carbohydrates are really very closely intertwined in what makes a native protein. And what we'll see, I'll show you the relationship between native protein architecture and actually features within the carbohydrates themselves. So we've been interested in this really in designing immunogens for HIV. And when, of course, the pandemic started, we were quick to sort of jump on and look at the coronaviruses in more detail. Now I show a sort of wolf in sheep's clothing here, because we can ask, why are these sugars here in the first place? And well, it's intimately linked to a couple of things. One is protein folding. So they're involved in the sort of calnexin, calreticulin mediated, chaperone mediated folding in the ER. But what's also driving such a high degree of glycosylation, we think, is predominantly immune evasion. So where you get viruses that are hanging around in the immune system under antibody selective pressure, you get quite a high degree of glycans appearing. Now you can see where coronaviruses fits on the spectrum here. We've got Lassa fever on the left, Ebola, flu, HIV, and coronavirus. And you can begin to see just how much of the surface is dominated by the sugars. Now, it's not just antibody immune evasion strategies that are sort of driving glycosylation. We also have a large number of different aspects of viral biology, including interacting with host lectins, all the way to sort of mediating the trafficking of viruses. Now, in this diagram, I really show, and it's a bit complicated, but there's a color scheme here I wanted to introduce, because N-linked glycans, the one I just showed you in blue, they get put on as oligomannose, which for our purposes, I'm just going to show as green. And as proteins mature through the Golgi, there's lots of additions, salicylic acid, galactose, and that's shown here in magenta. Now, viruses, because they've got so much sugar, they often overbake this sort of overbake the sugars, and that maturation process isn't always complete. And I'll show you a couple of examples of that. So why are we interested in recombinant systems? Obviously, it's critical for serology work, and some vaccine platforms are, of course, based on recombinant work. And I mentioned that we can, the sugars can influence how the protein behaves in the immune system, how it's trafficked. It can really be indicative of the underlying protein structure. In HIV in particular, you can get glycan epitopes. So that's where we have a particular focus on that. There's even some examples of antibodies recognizing carbohydrates in coronavirus, but That's rare. And also it can indicate, when you understand the sugar processing, the degree of shielding on your protein. So looking at HIV, and what we've learned there is if you take a GP120 monomer, or a sort of artificially trimerized pseudotrimer here we call it, or a native light trimer, the amount of oligomanos here shown in green, this is HPLC, is only really elevated when you get the native like trimeric structure. So there's protein directed reshaping of glycan processing. Now, we've been studying this site specifically using mass spectrometry, and in the HIV context, we basically set up and demonstrated we got pretty good quantitation. And of course, when you do online, almost like proteomics approach, you can start to see the sugars across each individual site, here it's shown on the right. And again, green is the mannose component, and magenta is the more matured complex glycans. Now in HIV, we can plot that on site specifically and show just how the structure of these sort of carbohydrates can cluster together, form these sort of oligomannose patches together with hotspots of more processed glycans. And when we compare that same methodology to comparing these monomers and trimers, you can see how trimerization is driving increase of oligomannose across the interface. So again, this really hammers home the interaction between protein folding and glycan processing. Now, one of the sort of slightly confusing things is I mentioned that these sugars can be susceptible to recognition by the innate immune system. Well, whilst being resistant to enzyme processing. And just quickly, you can see on the far left here, a picture of the mannosidases chewing away the sugar, and they really grab around the whole structure. Whereas the lectins here, shown in the middle, the yellow background, just recognize the tips. So you can have steric protection giving rise to mannose, and yet those are still very available for lectin recognition. Now in SARS, we worked with Jason McClellan's group to get the constructs of the soluble ectodomain of the protein, which in our hands behaved beautifully. We applied that same methodology, understanding the site-specific glycosylation. Here's actually 3 different batches, including batches made by in Texas, some in Southampton, and you can see the agreement is really close. So sugars, although it seems complicated, there's robust signatures of underlying architecture is really quite a reliable indicator, and you see it's very conserved. And we can then take those processing states, remap them onto the structure, and in contrast to what you just saw in HIV, despite the fact there's loads of sugars, actually it's a lot more processed. So we published one paper talking about the extensive vulnerability in the glycan shield. You know, frankly, these big enzymes have quite a lot of extensive access in amongst the sugars, whereas there's only a few hotspots like this N234, which is sort of based between domains and is sterically protected. We can put that amount of mannose on a more sliding scale here to show that there's still green mannose parts, but also there's sites which are mixed. They're still presenting some mannose, even though they're alongside complex. When we put that alongside other viruses, so back looking at those same ones I started with, you can see that the coronaviruses there shown on the left, again, show much more extensive processing, much fewer oligomannose structure, and generally much more accessible compared to Lassa fever on the right and HIV. So that correlates with basically vulnerability to antibody neutralization. So when we saw the sugar data, we said this coronavirus is going to be readily neutralized by antibodies quite extensively, unlike Lassa fever, which can be a bit problematic in terms of eliciting antibody responses to it. Now, we've been lucky enough to be given quite a few different samples from across the globe, really, of recombinant proteins. And here's just shown a small panel. And you can see basically just by eye, it's highly conserved, despite differences in CHO production here versus transient, you know, students making it, postdocs, even commercial entities, are all very similar, which is quite reassuring that if your protein is in the right, if you've got the right construct, labs across the globe are producing very similar type of material. We've also been lucky enough to work with Sai Li in China, looking at the glycosylation of actual viruses. Now, coronaviruses bud into the secretory system, so it wasn't immediately obvious whether you'd get normal processing similar to recombinant protein, but you can see comparing panel B with the recombinant average above, that by eye it's very close very closely recapitulated, which is fantastic news for both serology and actually for vaccines based on the recombinant glycoprotein. And finally, we've also been looking, and this is where the intersection between serology and vaccine design really shows here, is that we can look at the processing, the glycan processing of some of the RNA, or in this case, the ChAdOx vaccine. What's the quality of the material produced? And again, despite the fact you're impacting the cells with adenoviruses and doing all sorts of potentially strange things to your cell, it undergoes pretty normal processing. So we're presenting pre-fusion structure with glycoprotein that's undergone pretty normal maturation, in the sense that there's processed ones alongside steric. It's a bit different, but it's in the same ballpark. And interestingly, this is without using the stabilizing mutations that are common in the recombinant material I showed. So that's really quite reassuring for that vaccine format. That leads me to wrap up and thank everybody who contributed to this. There's millions of authors. When you look at the papers, I've pulled out a few people that dominated the work here, and in particular in the picture you can see Joel Allen and Yasunori. It's been doing a suitably socially distanced hello, and thanks for all the funders. So thank you.
Thank you, Max. That's a great talk. Well, I hope someone's going to show me some questions. Can I ask a quick question? Mm-hmm. So in inflammation, glycosylation of many proteins changes a lot. So during infection, early infection at home, and then you're admitted to hospital, then inflammation mounts even more as you end up in ITU. Does that change the glycosylation of spike protein within that individual and the way it interacts with antibody?
Yeah, I don't think we have evidence to address that either way, actually. So it's a good point. The whole area of viral glycosylation is really hindered by a lack of knowledge of the sort of state of the virus in vivo during infection. So almost invariably, people are looking at other allied features to indicate that. In HIV, we're lucky enough to have a large panel of antibodies that's specific to carbohydrates. And and some of them are even sensitive to the precise processing states. But even with those resources, it's quite difficult to actually characterize that, because you usually end up propagating your virus in order to test neutralization, and you lose the opportunity. So I'm going to have to just take a pass on that one. It could well vary quite considerably in detail. Now in the talk, I focus on monos versus complex. Where you can get variation is within the complex population, and that will be sensitive to all the things you're talking about. Whether that matters, I mean, I don't know, but I think it'll be a fruitful area to look in the future.
That's great, thank you. So, Lucy, I'm looking in the question boxes, and it's probably my incompetence, but I'm not finding further questions. So perhaps we should move on to Professor Alex Richter, who's going to talk to us about developing antibody assays and applying them in clinical practice across the whole spectrum.
Thanks very much, Mark, and thank you to Lucy and everyone for organising today. I hope everyone can see that. So today I'm going to just run through very quickly what we've been doing from antibody testing, really from design using Max's trimeric spike protein through to delivering and using these antibody tests in clinical practice. So remember back, it's a year ago now, so it's very good timing for this conference. We had a new virus, large numbers of patients. We didn't know who was vulnerable. We didn't know who had the disease. We didn't even know about anosmia. Had we already had COVID and dodged this bolus of severe disease? We had only very limited PCR testing and no community testing. So most people that had been unwell during this time, they didn't know whether they'd had another illness or whether they had had COVID. So in this environment, I set up a study called COCO to look at convalescent immunity in COVID, actually in healthcare workers. So this was a seroprevalence and a risk factor observational study. But importantly, it was a way of offering an antibody test to staff. Also, clearly, there were questions that I wanted to answer around the quality and the longevity of the immune response. Now, the first generation of antibody tests that we were going to use back in February and March and that we'd ordered in, we realized really early on that they lacked sensitivity. And the question was why? And what we worked out is that the assays were optimized for patients with known COVID from hospital. So the only patients we really knew about at the time were those that ended up with COVID pneumonia, were really unwell and on ITU. And actually, we didn't know about mild disease and that this could just be a transient phenomenon or even asymptomatic disease. Why is that important? Is because we found that the quantity of the antibody response varied. Now, a typical ITU patient would have significantly higher antibody levels than a symptomatic patient with mild disease or an asymptomatic patient. And, you know, given that over 85% of individuals do not have severe disease, it meant that the original assays were probably not fit for purpose, as they weren't looking at the limited detection for the target population. So we went to core principles and we started to build a team to build a new ELISA. This is where we worked with Max, and we also worked with other members of this group and in developing nuclear capsid and RBD antibody. And what we did is we tested in a kind of parallel way all the variables in every combination straight off. Not the usual way we do research, and certainly not the cheapest, but it was the way that it was the quickest way of getting the answers that we need. So we varied our antigenic target. We varied obviously the different type of sample matrices. We looked at serum and plasma, and we've done a lot of work with saliva as well, which I won't present today. We looked at the different detection antibodies, so monoclonal versus polyclonal, and we looked at combinations of G, A, and M, or all together. And then we varied various other just factors associated with an assay, like the buffers, the plastics, the incubations, etc., etc. And we found that the trimeric spike gave the best signal to noise ratio. So we were best able to differentiate between mild disease patients and pre-2019 negative controls. Certainly, RBD was fairly similar to the spike, but just not as consistent. And we found that there was significant overlap with nuclear capsid. So whilst nuclear capsid is a very abundant protein, actually we were getting an awful, we weren't getting good differentiation with the 2019 negatives. So we decided on the spike, and we were mostly running this on saliva. Looking at large numbers of samples with G, A, and M, we did find there was a remarkable amount of IgA. And so we hypothesized that if you combined G, A, and M together, we would be developing a more sensitive test. And if I just go through to describe that, we found a significant proportion could only be detected on a combined assay because the amount of G or A or M was just not enough to be detectable. So sensitivity analysis meant we went for this combined assay so we could pick up the very early M response and then the later class switch G and A response. And the other thing that we realized is that the original PCR testing had limitations. It's not a 100% sensitive test, and people with classic symptoms of disease were testing PCR negative, and we were picking them up with a sensitive antibody test. So in terms of a seroprevalence study, we now had an assay that was sensitive enough to be detecting people who had mild and even PCR negative disease. You might wonder about specificity, but by this point we'd run something like 800 pre-2019 negatives, so we were fairly sure of where our cutoff was. So looking at our in-house ELISA, which was now trimeric spike, it was a combined GA&M secondary antibody with a qualitative assay because we didn't have appropriate standards at this stage in the pandemic. We were finding 98% sensitivity and specificity in mild disease. And so this is when we went with to the binding site as a commercial partner to develop our assay for launch. And as with all things COVID, from conception to CE marking and launching took just a mere 4 months. So it was quite a pace. So how did we use this assay? Well, the initial results from COCO all came from a crazy 20-hour recruitment period just at the end of the first wave of the pandemic on the 24th and 5th of April. So we recruited about 516 members of staff from University Hospitals Birmingham. We had some really key findings. We established asymptomatic carriage in healthcare workers from PCR testing. And this was something that certainly our exec board wasn't aware of. But more than that, we'd found that 17% of our healthcare workers had already asymptomatically seroconverted. So actually the scale of asymptomatic transmission of the virus became important in the healthcare setting and obviously in the community. We confirmed that asymptomatic seroconversion was associated with lower antibody levels, and that becomes important when looking at the longevity of the immune response, because it's these lower levels that tend not to be maintained over longer periods of time. And I guess the key finding from this was that healthcare workers were at significant occupational risk from COVID, and a quarter of our healthcare workers were antibody positive after the first wave, which was much greater than the general population, which was only about 1% at the time. We found that higher antibody levels were associated with more systemic illnesses. So not just those with more severe disease, but those that had fever, more severe fatigue and diarrhea. And then we also found that age, ethnicity and BMI drove higher antibody responses. And this was interesting because we're starting to get a signal, weren't we, in the early summer that the severity of disease in older adults, those of Asian and Black ethnicity, and those with a higher BMI had a more severe disease. So it is interesting that even in mild disease, these patients were getting higher antibody levels. So our conclusion from this, and you know what we're trying to work through, is that really this implicates significant immune dysregulation in the pathogenesis of COVID disease. We all know you need an IgM antibody response initially, and then you get class switching and germinal centers, and you get an IgG and A response. And that peak in antibody response is usually about 6 to 8 weeks post-natural infection or vaccination, and then this can then be boosted. But after the antibody peak, it's perfectly normal for those antibody levels to wane. For many people, they remain positive, but for a number of individuals, that will drop below the limit of detection. Despite what the public, the press say about longevity of antibodies, it's not the whole story, is it? Because of course we've got memory B cells that can be reactivated to produce more antibodies if re-challenged, and there are T cell responses, which the next session will be addressing. But it is a marker of long-lived immunity. So we do look at how long the antibodies last for. And in a couple of very large cohorts, in the COCO cohort and also in a dental cohort that we've recruited called CovDent, we found that 2/3 of individuals remained antibody positive at 6 months following natural infection. And this fits with a number of studies internationally. So are these antibodies relevant or important? And what we found from our dental community, the timing of their 6-month bleeds was actually after the onset of vaccination, and we were able to look back over the 6 months as to what's happened to these patients and then relate that to whether they were antibody positive or negative at enrollment. And what we found is that antibodies afforded about 79% reduction in the risk of reinfection at 6 months. And I think this maps really nicely with what we're finding epidemiologically and in the press, is that we're not seeing huge numbers of reinfections, but of course we are seeing some reinfections. And the reinfections that we found in this cohort were mostly those associated with the lower antibody titers. And we found that those lower titers following often asymptomatic infection were lost or below the limit of detection on our assay at about 3 months. We've also put our antibodies into Vero neutralization assays and find that not just the baseline, but the 6-month follow-up antibodies show good neutralizing activity in vitro. So very reassuring. And then the last little bit to tease you all with is what's happening following vaccination. Because the assay is based on the spike, and of course all the vaccines are spike orientated, this assay is perfectly set up to look at the prime and the boost that we see with vaccination. And if you have a little look at these 2 sigmoid curves, the blue line is those that are infection naive. And you can see that most people are starting to get antibodies by 10 to 14 days, which is very reassuring. And it shows really some reasonable evidence that you can get protection even after the first dose. But what we found fascinating is if you look at the pink curve, these are individuals who have had an infection, so they've had natural infection. They can be boosted by 4 to 6 days. So a really, really early onset, rapid escalation in antibody concentrations. So all this natural infection information, I'm hoping will then get fed into the vaccine studies going forward, and also allow us to plan what we should be doing with booster vaccinations in the autumn. So just a huge thanks to our translational immunology collaborative team through basic science, clinical science, our organizational partners, and obviously our commercial partners. So thanks very much for your time. Any questions?
Alex, yes, there are questions. Thank you very much for that great talk. The first question is from Natasha Marin. Can you speculate why higher antibody levels are observed with certain ethnicities?
We actually don't know, and it's something we would love to know. I mean, there are a number of genomic studies going on at the moment, aren't there? Looking potentially at antigen presentation and whether that might be relevant, and we're hoping that something may come down from that. We do know in certain ethnicities that immunoglobulins tend to be a little higher anyway. So certainly in sub-Saharan Africa, often individuals have a higher IgG. And I think we've never known whether that is the environment or the individual that has driven that. But I think the findings from COVID are going to have wider legacy implications to try and understand this and the consequences of these differences.
Thanks. And from Letty, wondering, do you have any information linking antibody titers to long COVID?
No, not yet. And actually we are, we've got a number of long COVID cohorts coming through now, and it's a real mixed bag. But just add, there are various things with long COVID, I think, immunologically. So one thing I'm interested in is in the antibodies and how the antibodies may be interacting, obviously, with tissues and activating the immune system. And with that in mind, we've found a number of tissue-specific autoantibodies that potentially may have relevance in causing some of the more profound symptoms of long COVID, some of the neurological manifestations. So yes, again, early days, but watch this space. The cohorts are being set up now.
Good. And from Ella Mesami, what does the antibody yield look like in those who haven't got prior COVID? I guess she's thinking...
Is this a specificity question?
Yeah, I think this is what's the antibody yield look like in someone who's not had a previous COVID infection in response to vaccination?
Well, with in terms of cross-reactivity with other coronaviruses, we found with the trimeric spike protein, there was virtually no cross-reactivity with other coronaviruses. So our pre-2019 individuals were, universally negative. There was some story, I don't know if anyone heard it, about cross-reactivity with dengue. And so we got a whole load of dengue serum in to try and test them on our assay. And unfortunately, we didn't manage to pick any up. You do see the odd one that is cross-reactive, but on the whole, they are absolutely negative. And in terms of the antibodies generation in infection naive individuals, you are getting really good levels of antibody if you have been infection naive with vaccination, but quantitatively that's much, much lower than those that are boosted post-infection. Enormous levels are coming through.
So Alex, you'll be pleased to know that we're ahead of time, so we're going to get even more questions for you. So what is the lowest level of asymptomatic antibody detection quantity-wise?
So we're just working this out at the moment. So the NIPSCI and the WHO calibrators have really only been launched, well, it was about 17th of December. So we're just getting them into practice. And what we're doing is now building a quantitative IgG assay, and we're standardizing against the WHO standards. So we're also then looking to see where that cutoff point on the standards relates to our neutralization assays. So if you give me another couple of weeks, I'll be able to answer that question better.
Good, good. And final question. After receiving the vaccine, does showing symptoms prove the vaccine is working?
So I tend to say to any patient of mine that comes in who's had a significant vaccine response, I say, that's brilliant news. That means that you've produced a really good immune response. Now, in terms of what that response means, for the Oxford vaccine, there is a chance that you are making an immune response against the adenovirus, because some of us have seen, obviously, adenoviruses in the past. And indeed, the side effect profile with the adenovirus with the Oxford vaccine does seem to be slightly higher than the mRNA vaccines. But on the whole, if somebody is unwell and has induced a fever-like response, I tend to reassure people that they are making an immune response, and that's great news.
That's great. Thank you very much, Alex. We got our money's worth there. Not that we're paying you anything, but anyway. Moving on to the next speaker, please. Alexander Tarr from Nottingham, who's going to tell us about different patterns of antibody response in severe SARS-CoV-2 infections. So, Alexander, over to you, please. You're muted, Alexander.
Yeah, no, thank you. Oh, right. Finally there. Can you see those? Is that okay?
Yeah, can I just say, Alex, you need to put it onto Alex Tarr, please, now, if that's okay. It's the spotlight. Thank you.
Yes, it's fine, Tigby. Just bear with me.
Just 2 seconds, Alex, while we spotlight you, if that's okay.
That's okay. I can start if you wish.
Oh, we
Dan, it just hadn't accepted your video, that's all.
Excellent, thank you. Well, thanks for the opportunity to talk about our research on antibody responses to COVID. I'm going to describe some of the work we've been doing in the past year, particularly with my colleague Paddy Ty, who's optimized a lot of these assays and developed a lot of it. I get the opportunity to talk about it, hopefully discuss some of the outcomes of the work we've been doing. And it's been really well introduced already, so I won't go labour it too much. But really, when we started this work back in March last year, there was obviously very little known. We didn't really know what the contribution of antibody responses were to outcome of disease. And so this was one of the first questions we tried to address. Obviously, as time has progressed and everything's progressed very rapidly, we're now understanding that vaccines are generating neutralizing antibodies. There's evidence that neutralizing convalescent plasma is useful, is therapeutically useful. So we were characterizing a cohort of patients that we were treating in Nottingham, in the Queen's Medical Centre, who all had very severe disease. And while not all were in ICU, they had differing outcomes of infection. So we started off by taking samples. I'm a virologist working in a clinical virology setup. So we started taking serum samples from patients who were routinely giving blood for tests during the March, April, May period of 2020. These are all pillar 1 tests. They're all PCR positive, RT-PCR positive for COVID. So they were confirmed infections that we were tracking. And we generated over 1,000 samples in this very short period of time. People were getting sampled regularly, and we sampled almost on a daily basis some people. There were different outcomes with our patient group. 61% of the people who were inpatients in the QMC recovered and were discharged successfully, usually after 2, 3, 4 weeks of being an inpatient. However, 39% of our patients died before they could be discharged. So we wanted to look at the quality of the antibody response in different outcomes of disease. So that's the first set of data I'm going to show you. But before we could get going on answering these questions, we had to develop an assay. Alex has talked about assay development for ELISAs. We decided to use the Spike S1 subunit. I don't know if you can see my cursor here, but the S1 subunit includes the RBD. It binds to the ACE2 receptor. We thought this would be a good target for detecting antibodies. We rapidly worked out an assay system using our OpenTrons robot, so we all managed to automate the system. This allowed us to do very highly reproducible assays en masse, do hundreds of assays on these patients. So the very basic assay is you capture the antigen on a plastic plate, you use serum as the primary antibody, then use an anti-human FC conjugate, and then it's a basic ELISA system, very standard laboratory test. But done using robotics to increase the throughput. So we compared the anti-S1 reactivity to the anti-nucleocapsid reactivity. And the first question was to look at how rapid do these responses occur in our patient cohort. So instead of using the onset of symptoms, because we didn't have that data, we used the time from the first RT-PCR positive sample as our day 0. So people came into hospital, they were already ill, so they'd probably been infected for a week or 2, and then they had a PCR test. We used that as our day 0. As you can see, people were generating antibodies very early in infection to both to the S1 protein and the nucleocapsid protein. But it was intriguing to us that some people did not mount an antibody response to either antigen, despite being very poorly, despite clearly having COVID. They were prolonged infection with very low antibody titers. So it wasn't quite what we were expecting. We were expecting to see an antibody response in all the patients. Remember, at this point, it was all the Wuhan strain. We were getting that transition period between the early introduction of the 614G mutations in our population, and that actually happened. That sweep started to happen through the sampling period. I'll talk about that a little later. What was also interesting when we were comparing the nucleocapsid response to the anti-S response is that we only really saw anti-S responses where there was already an anti-N response. So all the samples that were anti-S positive were also anti-N positive, suggesting that maybe the N, as has been described before, was a more immunogenic protein. There's a more rapid response to anti-N, and maybe the anti-S develops a little bit later. So we took our patient cohort, and we stratified them by the outcomes. The first thing that was striking was that the age of these groups, as you'd expect, there were differences in the age populations. So we found that people who discharged, there was a large cohort of younger people who did better. So we did some of the subsequent analyses only on a group of patients that were all in the elderly. We chose 70 years as a cutoff, an arbitrary cutoff. When we compared the ages between those two groups, the died and discharged, there was no difference in the median age. But it was clear that age did have an effect on antibody responses, as has been reported by other people now as well. As you get older, as your patients get older, the antibody response is less potent, particularly to the S1 protein. A lot of patients still manage to mount a response to the nucleocapsid. But it was striking that when we compared in our elderly age group, people who were discharged versus people who died, there was clearly a better, stronger antibody response to both S1 and N in patients who were discharged. So these were all in samples taken after 7 days after that initial RT-PCR test. So we were confident at that point that if people were going to mount a response, they would have done it by then. So there was a clear difference between the different outcomes. What we thought might be more interesting is whether we can predict the outcome from the antibody response as people were brought into the hospital. Was there any predictive power of the antibody response? So taking samples before 7 days, so in the first 7-day window since they came into hospital and had the RT-PCR test, we took samples from discharged and people who discharged, people who died. There was no difference in the sampling days when we compared the window. And we compared samples only taken in that 7-day window. And the same thing was true when we did this. So anti-S response, anti-S1 responses are higher, marginally higher for anti-S1, but much higher for the anti-N. So if you mount an anti-N response as a marker of a good quality immune response, you're more likely to get better and resolve infection and be discharged from the hospital. Now, at the same time, other people were doing similar assays comparing endpoint titers. And so we thought maybe we might miss something by comparing absolute ODs between different patients at a fixed dose of serum. So we performed endpoint titrations. On the left here is just an example of the data set we got. What was interesting in the two top panels here on the right is that we expected to see a direct correlation between the endpoint titration and the initial signal that we generated with a single dose assay. But it wasn't quite as simple as that. And in fact, you see different profiles depending on the patient. So some people on the right here, you can dilute out the serum, get a high titer serum when it has a high OD in the single dose assay, but others dilute out much more rapidly. So there isn't a direct linear relationship. So it suggests that there are different affinity antibodies in different polyclonal responses to these two antigens. Again, but when we compared the outcomes with the endpoint titration assay, we found quite clearly that the people who were discharged, who got better from infection, were much more likely to have a more potent antibody response to S1 or N. So it held out whichever way we did the assay. The other assay we were developing at the time, working with Nigel Temperton at Kent, was the viral pseudotype assay. We've got a long history of doing viral pseudotype assays for other infections. And so we started doing neutralization experiments. And what we expected to see, and this in the top left-hand corner here, we've got some of the representative data. We thought there would be a range of neutralizing potencies, depending on how much anti-spike antibody was produced. So very simply, you make pseudotypes by transfecting an HIV backbone and the coronavirus spike protein into some hex cells. They produce these pseudotype particles, which are Cat2 containment, so you can do the neutralization assays very easily. And then we infect Vero E6 cells as the target cells. And by mixing the pseudotypes with antibody preps, you can then look at neutralization. What we discovered, we didn't expect to see. While the majority of the serum that we used in these assays were able to potently neutralize entry, some of the serum actually seemed to enhance entry. And we repeated this assay many times at different setups, and it always was the same result. Some patients who are with severe infection appear to manufacture antibodies that enhance the infection, at least of these pseudotypes. We haven't yet done it on a wild type virus, SARS-CoV-2 virus. But what was interesting is when you compare the anti-S1 ELISA data with a neutralization assay, you see that those sera that broadly bind to S1 were much more likely to have a neutralizing phenotype to those that had a low anti-S1 binding signal. So there's something in there, another antigen or some other component of the virus, maybe it's the S2 part of the spike protein that's responsible for this phenotype. That's something we're investigating. Again, going back to our patient groups, we compared patients who were discharged with patients that died in our cohort, and the neutralizing response was clearly stronger, more potent in those patients that were discharged. And the majority of the enhancing antibodies were observed in those patients that died. These again are samples taken in the first 7-day window after RT-PCR test. For a few of the patients, we were able to get sequential time points. And so I've selected some examples of the different patterns of antibody binding to the two antigens and neutralization that we saw. And it was interesting to see that over a period of 6 to 8 weeks, we were getting strong signal in some patients that maybe dropped off. Neutralizing potency were very high to start with. So patient 1, patient 2, 3 and 4 are all patients who were discharged and recovered. Generally, we saw a more potent neutralizing response linked to a good antibody, anti-S1 and anti-nucleocapsid response in the ELISA. Contrasting that with a few patients, we didn't have too many deceased patients where we had repeat samples over a period of time because they died. But in those patients that we did, they either didn't mount a response at all, very low neutralization, very low binding, or these antibody signals declined very rapidly after coming into hospital. So there are clearly different patterns of antibody response depending on the patient. Now, I said that as we were sampling these patients, we found that some were infected with the 614D variant and some were infected with the 614G variant. So we got this data from the UK COG consortium data, which is running in parallel here in Nottingham, where there's a huge amount of sequencing going on. And we overlaid on the sequence data onto our binding and neutralization data. And it was quite striking that both for the S1 and the N protein binding signals were higher in the infections that were infected with a G variant compared to a D variant. Remember, the antigen here is the Wuhan D strain. So this is cross-reactivity is greater in those infections with the newly emerging variant, the 614. The anti-S1 didn't reach significance, but when we did the neutralization tests and compared them this way, which is anti-spike antibodies, we found there was a significant difference with patients infected with the G variant having more potent neutralizing antibodies. So as time's progressed, there have been more variants occurring, and obviously the variant of concern at the moment is the B117. So more recently, bringing this up to date, more recently we've been looking at binding to the variant proteins as well as looking at reactivity to the original Wuhan strain. So I'll take you back to this, the cartoon I've shown before. The variants are in the top and bottom panels here on the left. I've shown the 614 in red, and then the B117 is the variants where there are mutations in red and deletions in yellow on that crystal structure. And it's quite striking. If we take our patient sera, remember these samples are taken from March, April, May last year. If we take those samples and look for cross-reactivity, we can see very good binding to the 614G variant compared to the original Wuhan strain. But in this 117 protein, we see very little cross-reactivity or unique reactivity to 117. We see much more a shift here to the reactivity to the Wuhan strain. So while there are some patients that mount cross-reactive responses to both the 117 and the Wuhan strain, there are quite a few patients that don't seem to mount responses that recognize this new variant. And in the top right-hand corner, I've just shown that the patterns of reactivity, some patients react mainly to the Wuhan strain with very little reactivity to 614G or 117, B117. Others have cross-reactivity to all of them to different degrees. So there are clearly different patterns, and it'd be fascinating to unpick this and work out what exactly those epitopes are that are being recognized. If we compare the antibody binding to B117 with the neutralizing potential against the Wuhan strain pseudotypes, we can see that if they mount a cross-reactive response to B117, so that's a high value here on the x-axis, they will be potently neutralizing as well. Whereas in those that don't mount a cross-reactive response, the neutralizing potency is actually quite variable between patients. And so just to finalize, given we had this genetic data on the different infections, we've split this data into those infections that were caused by the D614 variant and those that are generated by the 614G. So we've got D versus G infections and their reactivity to different proteins. And so the patterns for both those types of infections for the 614G protein seem to be very similar. So on the top, at the right and the left, those patterns are quite similar. But it's quite striking that in those infections that were caused by the 614D variant, we see low cross-reactivity to B117. But in those infections with the 614G variant, we're seeing a little bit more cross-reactivity. So the 614G is the predominant mutation that was at the base of the B lineage of infections. And so it might be that we're getting lineage-specific infections. Obviously, we need to use a lot of these different proteins that are now available to look at lots of different variants and the impact of specific mutations on antibody reactivity. But I think the take-home message is that we've got patients that have different outcomes of infection with different polyclonal responses that we need to understand in much more detail. So just to summarize, we've generated a well-characterized sample set, which we're now interrogating the very acute specificity of responses. We found that more potent neutralizing and binding responses seem to be associated with better prognosis in severe infection in hospitalized patients. Many of the patients generated neutralizing antibodies, but it was striking that about 18% of our infections generated enhancing antibodies. So that would be interesting to understand the mechanism of that enhancement. And as I've said, lineage-specific antibodies appear to be being produced in some patients. And so we're using these assays now to support a whole load of studies going on at Nottingham and elsewhere. We use these ELISA assays to help with the recruitment of the AstraZeneca vaccine trial. We're, like Alex, we're doing a lot of screening of healthcare workers and their family connections. We've been screening University of Nottingham students since August last year using these assays, looking for antibody responses to see what the prevalence of infection is across our student cohorts. And we're also looking at other niche groups like high-risk pediatric groups, including renal transplant patients. We're working with Lindy on looking at the neutralizing antibodies that are produced by DNA vaccines. And we're also assessing therapeutic mAbs that are being made in the lab. And finally, the epitope mapping I've already said, but I think this is a key point that we need to understand not only our responses, but also responses in other patient cohorts. So it'd be really good to work with other groups to try and get other patient data from a wider range of patient groups. Thank you. Oh, just to credit the people that really did a lot of this work. So Paddy Tai here on the left, Richard Banovitz, who is a master of pseudotyping, sorry, who did a lot of this work, and the group in Nottingham. And we've been collaborating with the NHS Trust widely, Public Health England. We've worked with Tim Brooks, who helped us validate the assays, and Nigel Temperton for helping us set up the pseudotype assay in the first place. Thank you.
Thank you very much, Alexander. That's a fascinating talk and clearly some really exciting things for the future. We just got time for one very quick question. Were factors taken into consideration, such as different medicine interventions, I guess at that time, particularly dexamethasone or not, and the antibody response? Yeah,
we did look at that. There were so few patients receiving dexamethasone in our cohorts that we excluded those patients. So we looked at people that weren't treated.
Thank you very much. So I think here I better end our session so that we're timely for our organisers and pass over to Lucy, I think.